Hello friends, welcome back to the Inquisitive channel. In this video, we'll be talking about cell organelles, and specifically animal cell organelles. Now this video is a continuation of a previous video where we talked a bit about cells and actually touched upon cell organelles. So please feel free to go back and view that video first, although that's actually not necessary to understand the content in this video. All right then, let's get down to it. So cell organelles. Now, in terms of which organelles specifically we're going to talk about the main ones. Okay, so we'll talk about the nucleus, the ribosomes, the mitochondria, the cell membrane, the cytoplasm, the endoplasmic reticulum, both rough and the smooth type, the Golgi bodies, sometimes known as the Golgi apparatus, the lysosomes, and the cytoskeleton. Okay, so we're going to briefly go and talk about all these organelles. But before I do that, let me just remind everyone the sort of target audience for these videos, especially for these videos that I'll be discussing biology with. Now, the content for these videos is designed for students who are studying in the UK for their GCSEs, and high school diplomas in the US, and similar. I mean, I'm really talking about high school or secondary school education level. Right. Okay then. So now that we've got that over with, let's start off with the nucleus. Okay. So if you look at this picture here, this shows a collection of cells in a tissue. The nucleus is usually the biggest structure, the biggest organelle that you can observe under a microscope. Okay, so these cells have been stained with a blue dye to make sure that you can actually see the nucleus. Okay. Now, in terms of what the nucleus does, so let me give you some context. So sometimes context helps to explain things a bit better. Right then, so each cell has to perform many tasks. Yeah, there's many biochemical reactions that have to take place, and the cell has to make sure that these biochemical reactions take place at the right time, in the right quantities. Okay. Now, in order for it to achieve what it's supposed to do, okay, and fulfill each task, it needs a set of instructions. Okay. Now, in the cell, the instructions come in the form of proteins. So each protein has an objective. Okay. Now you can think of this objective as a word. Okay. A word that makes up a sentence of instructions. Okay. So imagine if there are words missing from a set of instructions, it will be difficult to understand those instructions. Okay. So similarly, if there are proteins missing, okay, the cell has difficulty to actually undertake those instructions then, or follow those instructions. Okay. So the instructions have to be spelled out. In other words, in the cell, the proteins have to be present at the right time, at the right quantity, and in the right format. So, okay, so when you consider the number of processes that happen in a cell, the number of processes can be in the order of thousands. Okay. So if there are that many instructions which are mediated by proteins, then you understand that the number of proteins that are needed to enable these instructions to be followed properly is also going to be in the thousands. Okay, so at any given moment, there are thousands and thousands of proteins expressed in a cell, okay, that are expressed at the right time and in the right quantities. Okay, and that is really important. Okay, so that's really important to get across. Okay, to get something produced at the right time in the right quantities requires a fine level of control, and this is what happens in the nucleus. Remember, the nucleus is the control center. Okay, so in order for the actual cell, for the cell to control proteins, proteins have to be produced and then they have to be removed. Okay, and so proteins do have a finite lifetime. In other words, once the instructions have been followed, okay, so once the actual aim of the protein is complete, that same protein can't hang around for too long. Okay, because the same proteins can then interfere with other instructions for other processes. Okay, and that can be quite dangerous. So for this reason, proteins are removed by one means or another. I'm not going to go by how they're removed, okay, but just be aware that they're removed. They can either be switched off or they can be destroyed. Okay, so what this hopefully demonstrates is that proteins undergo a continuous cycle of production and destruction. Okay, now in order for this complex process to go ahead in the right way at the right time, in the right quantities, there must be a control center that is overlooking all these thousands of processes. Okay, and like I said, that is the nucleus. Nucleus is the control center. The control center, in other words, the nucleus, is able to monitor the condition of the cell and respond very quickly by switching on certain genes or switching off certain genes so that the right proteins are produced at the right time and in the right quantities. Okay, so remember, genes code for proteins. Okay, so that's kind of the broader picture that I wanted to get across before I actually talked about the nucleus itself. Okay, so the nucleus, as we said, is the control center. Okay, this is what monitors and ensures that the right genes are being expressed, which means that the right proteins are being expressed, okay, at the right times, at the right quantities. Okay, now, okay, so the genes are located on chromosomes. Okay, now, in case you don't know what chromosomes are, okay, chromosomes are basically it's a massive length of DNA. Okay, a massive length of DNA with which is sort of split into segments. Okay, and each segment you could consider it as a gene. Okay, so genes are the DNA segments that code for proteins. And essentially, if this is a bit of a generalization, it says here one gene codes for one protein. There's two asterisks here. That's just to mean that it's not quite as simple as that. Okay, there are processes like alternative splicing and post-translational modifications that can give rise to diversity. So one gene doesn't necessarily give rise to one protein. Okay, it can give rise to protein, but then there can be variations of that protein. Okay, and this is mediated by processes like alternative splicing and post-translational modifications. Now, don't worry about these processes for the time being. Okay, as long as I've introduced them, you can go and look it up. But we might come back to these processes in a later video. Okay. Now, I wanted to talk about transcription here. Okay, we've talked a lot about instructions. Okay, now the source of these instructions all exist in the nucleus. Okay, a gene is basically a set of instructions that tell the cell to produce which protein. Now, what are proteins made up of? Okay, they're made up of amino acids. Okay, so it's a case of that gene encoding a set of instructions to make that protein, which is basically a set of instructions to say which amino acid comes first, which amino acid comes second, third, fourth, fifth, etc. Okay, and it's only then that that protein can be produced. Right, so the DNA of a gene serves as a template. Okay, now you can consider this as a kind of a blueprint. The blueprint exists in the nucleus. Okay, there's only one blueprint. So if you wanted to pass on the instructions in the blueprint, you'd have to make a copy of it. Okay, and pass it on to the other components of the cell. And this is what actually happens. Okay, a copy of that gene is made, okay, but not in the form of DNA. Okay, there's a slightly different version of DNA. Okay, it's called RNA. Okay, incidentally, if anybody's interested, what this stands for, if you don't already know, is deoxyribonucleic acid, and RNA is ribonucleic acid. So nucleic acid. So they're both nucleic acids. Okay. Now this whole copying process. Okay, so making a copy of a particular gene. This process is called transcription. Okay. Now, I wanted to spend a moment talking about transcription because transcription is actually part of the process to explain how cells produce or synthesize proteins. Okay. Now, this is the this is DNA. It's a double-stranded helix. Okay, and in order for what we have is we have two strands. We have a non-coding strand and a coding strand. Okay. Now, the DNA is it's a relatively simple molecule. What makes it complicated is simply the length of it. Okay. Now, to give you an idea how much DNA there is per cell, just per cell there is, and remember this is a cell that's invisible because it's so small. If you took all the DNA from a cell, okay, it'd be 6 feet long. Okay, that's just per cell. So you can imagine. So in other words, if you were to unravel all the DNA from all the chromosomes made into a single strand, it would be 6 feet long. Okay. Okay. So that's just to give you an idea of how extensive the DNA is per cell. Okay. So here we have the double-stranded DNA molecule, and what you'll find is that the gene is composed of what we call nucleotides. Okay, so A, T, G, C. These are the 4 different nucleotides. Okay, now this is how we represent a gene. Okay, the actual sequence of a gene, we describe it in the form of these nucleotides. So ATG, ATC, TCG, TAA. Okay, and incidentally, one amino acid is coded by a triplet. Okay, so this ATG will code for one amino acid. Okay, ATC will be another amino acid, TCG will be another amino acid, and TAA will be another amino acid. Okay, so this is how the order of amino acids is coded. Okay. So we're gonna okay. Let's get back to transcription. So in order for a copy of this coding strand to be made, there has to be some unraveling. Okay, so just to sort of open it up a little bit, and then we have a special protein called an RNA polymerase. Okay, and again, I'm introducing new molecules and new terminology. Don't worry about the new terminologies just yet. Okay, but it's just a special protein, a special enzyme, if you like, that is able to copy the coding strand of DNA. Okay, so it copies it. Okay, and remember, it's another nucleic acid, but it's a different type of nucleic acid. This is deoxyribonucleic acid, and the copied version comes in the form of ribonucleic acid. Okay, so it's copied and then it's transported. So remember, this is happening in the nucleus, and this is in the cytoplasm. Now let me give you some text here. Okay, so so a gene is a segment of DNA which is a double-stranded molecule. One strand serves as the coding strand. Okay, so in this case, it's this bottom strand here. That's the coding strand. Okay, now the double-stranded molecule unwinds, allowing a copy of the coding strand to be made. This is called transcription. Okay, so this process is transcription. And then what happens? Okay, that copy of the coding strand is transported out from the nucleus and into the cytoplasm. Okay, where it is decoded. Of course, remember it's a code. Okay, it's not anything can actually decode this. What decodes this is another organelle called a ribosome. Okay, so that transcript, if you like, the RNA transcript, is decoded by ribosomes to produce a protein. Okay, and when we talk about ribosomes reading the RNA molecule to produce a protein. That's called translation. Okay, so we have transcription, and we have translation. Okay, and both of them are required, okay, to complete the synthesis of proteins. Okay, so in the previous slide, I just introduced two things, ribosomes and translation. So let me talk a bit more about ribosomes and translation here. Okay, and look at the relationship between RNA, ribosomes, and translation. So it's ribosomes. So these are the ribosomes, okay, which actually sit on the RNA molecule. Now it says mRNA here. This M stands for messenger RNA. So the issue is, okay, there are different types of RNA molecules. Okay, so to differentiate the different RNA molecules, okay, to differentiate the one that is produced after transcription, we call that RNA molecule a messenger RNA. Okay, messenger RNA, or shortened to mRNA. Okay, now ribosomes are organelles, okay, that are used for protein production. Okay, and the process they mediate is a process called translation, which we introduced in the previous slide. Okay, it's these ribosomes that sit on the messenger RNA molecule and basically just read along the molecule. Okay, remember the triplet nucleotides, ATG, CAT, etc. Okay, so these ribosomes basically read and decode that information. So if ATG decodes for a particular amino acid, the ribosome will attach that amino acid. Okay, it'll go along to the next triplet of nucleotides, which will code for another amino acid. Okay, and the ribosome will then attach another amino acid, the relevant amino acid. Okay, and it'll keep doing this. It'll keep doing it, and eventually you get a long strand of amino acids. Okay, and then it hits the stopping point. Okay, and again there's a coding signal which tells the ribosomes that the gene ends here, so to stop here. So it stops there, and then that chain of amino acids is then released. Okay, and then the ribosomes are recycled then. Okay, so all these processes like transcription and translation and the multitude of other processes that happen inside the cell, okay, all require energy. Okay, energy is what drives all these processes to go ahead. So the cell must have a way to produce this energy. Okay, and we do have organelles within a cell called mitochondria. Okay, now there are hundreds and hundreds of mitochondria per cell, and their job is to produce one. They have other jobs as well, but their main job is to produce energy. Okay, mitochondria are sometimes referred to as the powerhouses of the cell, the power station of the cell. Okay, so the actual process of producing energy is quite a complex process. Okay, a lot of it happens in another part of the cell called the cytoplasm. Okay, but the energy yielding part happens in the mitochondria. So if you think about things that we consume, such as carbohydrates, one of the major carbohydrates we consume comes in the form of glucose, sugars. Okay, so it's these sugars which are broken down in the cytoplasm, and then the broken down products are sent into the mitochondria. Okay, where it's then broken down further to basically yield the energy. Okay, now this energy that's locked away in glucose, for example, and other molecules can also be used for energy purposes as well. Okay, but the main nutrient we use for energy purposes is sugars, carbohydrates, glucose, for example. Now, the energy locked away in glucose is harnessed, okay, and it's transferred to this molecule here called adenosine triphosphate. Okay, the energy is stored in this molecule of ATP, adenosine triphosphate. Okay, and processes that require energy use ATP. Okay, so the processes that happen inside a cell can't use glucose directly. There's lots of energy locked away in glucose, but it can't harness that energy. Okay, it has to come in a currency that the actual cell understands, and the currency of energy that our cells use comes in the form of ATP. Okay, adenosine triphosphate. Okay, uh, it's basically 3 phosphate molecules attached to this sugar called adenosine. Okay, and it's these phosphates that when they're attached to adenosine, okay, lock the actual energy into the molecule. Okay, and it's in the form of ATP that cells can actually use and harness the energy much easier. Okay, so to quickly summarize then, the mitochondria is the organelle which produces energy. Okay, very important. The next organelle that I wanted to talk about is the cell membrane. Okay, it's this membrane which encapsulates the cell. Okay, which holds everything inside. Okay, that's one of the actual functions of the cell membrane, is to keep everything inside. Okay, the cell membrane is what is made up of is what we call a lipid bilayer. Okay, lipid bilayer, which means it's basically made up of fat. Okay, now we're not going to really talk about the structure of the lipid bilayer. Okay, but just to say that this is important. Okay, the lipid bilayer is made up of several fats, but cholesterol as well. Cholesterol also plays a big role in the production of the cell membrane. Cholesterol has a bad reputation, but it is so important in so many different structural elements of the cell. Okay, it's also used for energy purposes as well. Okay, but cholesterol, one of the major uses for that is in the production of the lipid bilayer. Now, incidentally, okay, the cell membrane is made up of this lipid bilayer, but every organelle that we've mentioned and the ones we're going to mention yet are also made up of a lipid bilayer. Okay, they also have membranes, not cell membranes, but they have membranes which are made up of lipid bilayers. So a mitochondria is also has a lipid bilayer. So it's what makes them their own entities. Okay, so that's enough about cholesterol. One of the other, and it's actually a main function of the cell membrane rather, is that it helps to control transport of molecules in and out. Okay, the outside of a cell is quite different to the inside of a cell. Okay, the outside of a cell actually can be quite dangerous to the inside of a cell, so that's why it has to be protected, and the cell membrane protects itself from the outside through the cell membrane. Okay, the cell membrane also, as I said, helps to transport molecules in and out. Okay, we consume food, nutrition, that's all broken down, it's digested, and then those nutrients are passed from the digestion and into individual cells. Okay, so one of the nutrients which we've already mentioned is glucose. Okay, now glucose can't just enter the cell nilly-willy. Okay, there has to be a control mechanism by which glucose. The cell will only require so much glucose. It can't. If too much enters the cell, it can be dangerous. So the cell has to control how much glucose it actually needs. Okay, so the cell membrane facilitates this entry of glucose, not just the entry of glucose, but also the exit of other molecules as well. Okay, so there's this careful control of transportation in and out of the cell, mediated by the cell membrane. It's not quite as simple as that, because what you'll find is that the cell membrane itself doesn't actually do the transportation. There are what we call receptors embedded within. Okay, receptors embedded within the cell membrane that allow transportation of specific molecules. So on the subject of glucose, okay, we will have specific receptors embedded within the cell membrane that allow movement of glucose into the cell. Okay. Okay. So the glucose receptor is just an example of one receptor embedded in the cell membrane. There are hundreds and hundreds of others. Okay, and each one has its own function to allow transport in and out. Okay, so the cell membrane is actually important in allowing the embedding of these receptors. Okay, which then allow movement of substances in and out of the cell. Okay, so that's one of the main functions of the cell membrane, is to control transport of molecules that are coming in and out of the cell. Okay, so the next thing I wanted to mention was the cytoplasm. Okay, the cytoplasm. The cytoplasm is a very, it's not quite watery, but it's more like a gel type liquid. Okay, so it's quite viscous. And it's this cytoplasm in which all the organelles are suspended in. Okay. Now the consistency of the cytoplasm is important. Okay. If it's too viscous, movement of molecules to and fro becomes more difficult. Okay. Then if it's too watery, again that influences the movement of molecules. So the consistency of the cytoplasm is important, and this is when things like dehydration comes into play. If a person is dehydrated, the gel-like substance, the cytoplasm, becomes a bit more viscous. Okay, and that can be quite dangerous to cells. Okay. Another thing about the cytoplasm is that all the raw materials needed by the cells are also dissolved in the cytoplasm. Okay, so everything that the cytoplasm, everything the cell needs, such as amino acids, such as minerals, such as glucose, these are all dissolved in the cytoplasm. Okay, part of the cell respiration pathway happens in the cytoplasm. We talked about the mitochondria using the breakdown products of glucose to generate ATP. Okay, the actual breakdown of the glucose happens in the cytoplasm. Okay, so cell respiration, the breakdown of energy-rich molecules such as glucose, actually begins in the cytoplasm before transportation into the mitochondria for ATP synthesis. Okay, so the next very important organelle that I wanted to mention was the endoplasmic reticulum. Now, where is the endoplasmic reticulum on here? Okay, so it's this here. It's these sheets of membrane very closely associated with the nucleus. Okay, and that's important, as you'll see in a minute. Okay, but this is where most of the endoplasmic reticulum is very close to the nucleus. It's physically associated with the nucleus. Okay, there are two types of endoplasmic reticulum. One type we call rough, and the other type we call smooth. Okay, so rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, why do we call it rough and smooth? Well, what you'll find is that under a very good microscope, the appearance of the rough endoplasmic reticulum is because it looks rough, and it looks rough because there are ribosomes attached to those portions of the endoplasmic reticulum. Okay, and it's these ribosomes which give the rough appearance. And if you remember what the role of ribosomes was, it was to synthesize, to produce proteins from messenger RNA. So therefore, it makes sense, doesn't it? The messenger RNA, which is synthesized in the nucleus, doesn't have much to travel outside of the nucleus before it encounters the rough endoplasm, or before it encounters the ribosomes on the endoplasmic reticulum. So therefore, they can be synthesized quite quickly. Imagine if the endoplasmic reticulum or the ribosomes were outside, far away from the nucleus. The messenger RNA would have to travel to those ribosomes. That's not to say ribosomes don't exist in the cytoplasm. Independently, they do. Okay, but the rough endoplasmic reticulum, okay, helps to mediate quick synthesis of proteins through messenger RNA delivery to the rough endoplasmic reticulum. Okay. Okay, so this is the site of active protein synthesis because of the ribosomes attachment. Okay, so messenger RNA is transported from the nucleus to the awaiting ribosomes attached to the ER, to the endoplasmic reticulum. Okay, so once the actual protein is synthesized by the ribosomes, okay, the smooth endoplasmic reticulum portion of it, okay, that facilitates the correct folding of protein. So it contains it contains an environment that helps the proteins to be folded properly. Okay, correct folding of proteins is so important. Okay, because if proteins don't fold properly, in other words, they're misfolded. Okay, as soon as they're misfolded, the cell senses that and targets them for destruction. Okay, so the correct folding of proteins is important, and it's the endoplasmic reticulum is also involved in the production of proteins. Okay, there are other functions of the endoplasmic reticulum as well. Okay, but we'll stick to that for the time being. Okay, so once the proteins are properly folded, okay, they're assembled, they're correctly assembled in the endoplasmic reticulum. Once that's complete, okay, these proteins, and sometimes they're protein complexes, in other words, that are multiple proteins sort of joined together, okay, are they're then transported from the endoplasmic reticulum to the Golgi bodies, okay, which is the next organelle that I wanted to talk about. Okay, now the Golgi bodies plays plays a role in transportation. Okay, it plays a role in modification as well a little bit. Okay, so correctly folded proteins transported from the endoplasmic reticulum are received by the Golgi bodies. Okay, and the Golgi bodies are involved with modifications. Okay, so when we talk about, I know I mentioned this before, post-translational modifications, and this is where we can add other components to proteins. We can add sugar molecules, we can add fat molecules, and we can change the function of those proteins by adding these molecules. So that's called post-translational modification. Okay, and this happens in the Golgi bodies. Okay, in terms of transportation, okay, the Golgi bodies, you can think of an analogy as a post office. Okay, whereas a post office makes sure that mail gets to the right address. Okay, the Golgi bodies ensures that proteins get to the right places, not only within the cell but also outside the cell as well. Okay, so if something is produced and it needs to, say a protein is produced and it needs to be delivered somewhere over here. Okay, the Golgi bodies ensures that that protein is delivered to the right place. Okay, so it's kind of a sorting place as well, where and the way it does it attaches molecules to the proteins, which are then used for delivery to the right places. Okay. I did mention that we can add, the Golgi bodies can add sugar molecules to the proteins. When we're talking about adding sugar molecules, we call that glycosylation. Okay, so this is important for many proteins. Okay, and simply by adding sugar molecules to those proteins, it can give the proteins an alternative function. Okay, so this is when I was saying that one gene doesn't necessarily code for one protein. Okay, sort of in a general sense it does. Okay, but the proteins can be modified to have a different function. Okay, so this is the penultimate organelle that I wanted to mention, and those are the lysosomes. Okay, now the purpose of the lysosomes is actually in the title, lyso. Lyso meaning lytic, meaning breaking down. Okay, now in the picture of the cell here, okay, so you can see the lysosome is represented within this blue, purple rather, circle here. Okay. And like the mitochondria, like the Golgi bodies, there isn't just one copy of it. There's multiple copies of lysosomes within the cell. Okay, so lysosomes contain lytic enzymes, and the purpose of lytic enzymes is to break down, in this case, organic polymers. Okay, so proteins are examples of organic polymers. Nucleic acids are as well, such as DNA and RNA. Okay, and carbohydrates and lipids. These are all examples of organic polymers. Okay, so anything that's unwanted, anything that's been used up is gone beyond its shelf date. It's then transported into the lysosome, where these lytic enzymes get into action and break these substances down. Okay, so you can think of lysosomes as also recycling centers. Okay, they break down proteins into individual amino acids, and then those amino acids can be used to produce helped to synthesize other proteins. Okay, nucleic acids are broken down to help synthesize new DNA and RNA perhaps. Okay, and carbohydrates are broken down to synthesize the building blocks of carbohydrates, which are glucose molecules. Okay, and fats are broken down into lipids. Okay, fatty acids. Okay, so that's the purpose of lysosomes. Okay. Now there are special cell types. I just want to add a little bit more, but there are special cell types. Okay, so white cells, for example. Okay, white cells which fight off bacteria. They basically engulf bacteria. Okay, and destroy them once they're engulfed. And it's these lysosomes which help destroy those bacteria in those white cells. Okay, so every cell will have lysosomes inside. Okay, but then some cell types, like certain white cells, will have a lot more lysosomes because they have to kill bacteria. So here we are, folks. We got to the end. The cytoskeleton. This is the last organelle that I wanted to talk about. Okay, so the cytoskeleton is really important for transportation. Okay, so you can see up here that the cell actually has a very fine meshwork of fibers, okay, made up of proteins. Okay, and it's this meshwork that gives the cell its actual structure. Okay, now they're not fixed. This is quite dynamic, so they're constantly changing. So that's where the cell can actually change its shape, and cells do change their shape. Okay, and it's mediated by the changing of the cytoskeleton. Okay. So embedded within the cytoplasm, like I said, is this meshwork of protein filaments or fibers that make up the cytoskeleton. Okay. It's important for maintaining the cell shape and internal organization of the organelles as well. Okay. It is used to provide a network for transportation of organelles and other molecules. Okay, so if something needed to actually get from here to there, it would follow the path of the cytoskeleton to get to its destination. It's not free to move entirely by itself. There is a sort of this almost invisible network of of fibers. Right then. You can start to see the cytoskeleton in action when you see the cell divide. Okay, so when a cell starts to divide, you can see that this is the chromosomes. Okay, so this might be the parent cell, this might be the daughter cell. Okay, so the parent cell has its chromosomes and the daughter cell has its chromosomes. Okay, to begin with, they're both together. Okay, but when the synthesis of the new chromosomes is complete, it is time to pass on the new chromosomes to the daughter cell, and you can see that there's a certain pattern. Okay, there's a certain pattern to this to how these chromosomes actually arrange themselves, and it follows the same patterns of cytoskeleton. So it uses the cytoskeleton to actually facilitate movement. Okay. So the cytoskeleton ensures that cell division takes place appropriately, okay, whereby chromosomes are shared equally amongst parent and daughter cells. Okay, so the cytoskeleton, very important in terms of transportation and maintaining cell shape and internal organization of organelles. Okay, so that's it for now. I'm sorry the video took so long. I didn't want to break this one up because I thought it's better to have all the organelles in one video. So it has taken a bit longer than I would have liked. I know concentration spans are not that long for everyone. Okay, the sort of average concentration span is about 20 minutes. Okay, so it's whether you stopped, paused it, and view it for later. Okay, whatever suits you better. But thank you for watching this video. I hope it has been useful. Okay, I hope it does prepare you for or gives you a more broader understanding of GCSE level biology, at least in terms of cells and organelles. Okay, so as always, please leave a comment or post a question. I will respond to questions if you do have any. Okay, and like I said in my other videos, if you do want to see more videos like this, if you do want, please hit the subscribe button. Okay, it's only through subscription that I get an understanding of, I get an idea of how many people are actually interested in the videos that I make. Okay, or if they're not interested, then it gives me an idea of what I need to change. Okay, but comments and subscriptions really important. Okay, thank you. Goodbye. Until next time. Okay, stay safe, everyone. Bye bye.